the last speaker of the session is going to be Brendon Watson from University of Michigan. His talk is going to be recording across the neocortical layers in mouse S1 during task performance and sleep. And of course, we are looking forward to a robust discussion panel afterwards. So Brendon, it's all yours.
All right, thank you, Dr. Jung. Can you guys see my slides and
Yes.
hear me? All right. So I am rolling the dice. I am at home. And if you hear violin sounds, that's a real 10-year-old. You're not hallucinating. Anyway, so I'm a psychiatrist, but I do all rodent research. And so this will be a little bit of a departure from some of the other talks. It's not so neuroengineering oriented, but it is oriented towards some of the basic structure function of cortical circuits. And so I think it can be instructive to a lot of the efforts that were discussed here and that may be discussed for the remainder of the meeting as well. So just as an overview, I wanted to kind of start with some of the way we're thinking about the cortex and the tools that we're using. And I wanted to make this a little bit more of a tech-oriented talk because of the fact that this is NeuroNexus and that we're beginning to look at how cortical function correlates with performance. And then I'll kind of have to switch gears just because of COVID. We ended up stopping some of our experiments, but we ended up discovering something new. And so the second half will be using some of the same recordings. So first I'll sort of tell you where we're planning to go and some of where we're starting to see with the first set of data. And then I'll switch gears and tell you a little bit about how sleep is functioning in these same recordings. So just so you'll not be too surprised when there's a little bit of a change of gears there. Okay, so the neocortex is made of layered neurons, right? So this is I hope you can see my arrow cursor. This is from Cajal over 100 years ago. This is just a drawing showing that in certain parts of the cortex, especially, there are very clearly different layers to the cortex with different densities, for examples of these pyramidal cells that he's drawn. And where modern neuroscience has spent a lot of effort to try to build kind of a network diagram of these cortical circuits. And so there are, of course, 6 layers, really kind of combined 2 and 3. So layer 1, 2, 3, 4, 5, and 6. And so people have done a great deal of work to try to map out connectivities across these networks. And a major way of studying these has been by using the barrel cortex, which is basically the primary somatosensory cortex in rodents. The whiskers are very important in rodents, and so the whisker system is part of the somatosensory system, and it's very developed in rodents. And it furthermore really gives us nice abilities to look at, let's say, the input from one whisker and map it to one region of cortex, and the input to another whisker to another region of cortex. And then it has very nicely delineated layers 1 through 6 as well. So it's been a model system, and that's what we're going to be looking today. And there's this idea of cortical columns, which is also really nicely highlighted in the barrel cortex. These columns, and this is drawn by, these are from Mountcastle, Vernon Mountcastle in an older review, showing that each of these columns, these kind of horizontally tiled regions of cortex, each has a series of layers throughout its depths, but each may be dedicated to something individual, to different kinds of information, right? And as we heard earlier, this dedication and mapping is certainly not as simple as it sounds, but there is some tendency. For example, Hubel and Wiesel found that as they went across these columns, neurons tended to fire in response to certain kinds of stimuli, let's say lines at certain orientations in the visual system, versus in another column, they might respond to lines of a different orientation. And in the barrel cortex, in the barrel system, you can actually see these columns really nicely, even with simple stains. And each of these is dedicated to one whisker. And again, actually they share information across whiskers, across columns, but for the most part, the wiring is most strongly dedicated coming from the thalamus, from single whisker to the cortex. And so it's been a really nice model system. And we're sort of intentionally, so like I said, I'm a psychiatrist, and there's a whole part of my lab that's looking at strats and the prefrontal cortex. I intentionally went back to this well-known, well-studied structure. It's actually what I did my PhD in, because I want something tractable. And so this is about as tractable as it gets for a cortical physiologist: visual cortex, somatosensory cortex. So with the knowledge that there are certain caveats, but with the intention of going into certain kinds of depths that one has to use a well-known system to go into, we're going to be using barrel cortex. So lots of work over the years has been done with patch clamp and with other neuroanatomical mapping methods to figure out a basic cortical network, right? So a wiring diagram. Information from the thalamus comes to layer 4 in the sort of canonical classical circuit. These layer 4 neurons then send inputs to layers 2, 3, which then send inputs to layer 5. And so this is found many ways, using many methods, to be this kind of canonical circuit. And again, that this is part of these kinds of circuits, are some of what cortical anatomists have been trying to map out for years, to try to understand, how does a cortex function? How does it process what it processes? How does it let you move? How does it let you perceive? How is it that, there may not be actually orientation selectivity. There may not be only firing to certain kinds of orientation in layer 4, but Hubel and Wiesel found that in layers 2, 3, and 5, there is orientation selectivity. How does that happen? What is this column doing, this column made of layers doing computationally? So it's a very crucial kind of to certain assumptions of how the cortex works to understand how these layers work together. But it's been difficult using some of the more popular methods lately, for example, two-photon microscopy, where people do really nice work these days looking at tasks like go, no-go tasks or other performance behaviors. Those are spanning horizontal tracts of cortex, right? They don't cut vertically. And so it's actually, despite the importance that we think these layers have, it's been hard to study them systematically and simultaneously. And in fact, it turns out that some of these assumptions about this canonical circuit are probably wrong. That there's not just one circuit, it's not just kind of completely dictating things. So here's an example from Ken Harris's lab. They gave a sensory stimulus, in this case an auditory stimulus, and showed that activity started in layer 4, and actually in these deep layers as well. There's actually another thalamic input to the deep layers, but kind of, right, so I'm already breaking this canonical circuit, and that's going to be one of my points, right, is that this canonical circuit isn't quite as well established as we think in the cortex. It's not the only thing. So, but in this case, we give a sensory stimulus. The spiking is what's shown here in a raster, starts in layers 4 and 5, 6, where there are thalamic inputs, and then it spreads outward over time, right, to layers 2, 3, and 5. But basically in either anesthesia or in sleep, we have these upstates or delta waves, and they can start in layer 5, for example, instead. And I actually noticed that Richard Fyeth is in the audience, and he has some really nice findings showing even these upstates can start in different places, different layers, right? And so things can propagate lots of different directions. And so this canonical idea that inputs come to one place and then spread in this very canonical way is almost certainly too simplified. A more accurate version of things is probably that there's kind of all-to-all connectivity, and that these canonical circuits are just a bit stronger than all the rest. The lines are just a little bit thicker than all the rest. And in fact, a lot of the computational and modelers looking at cortical circuits will actually sometimes model them as all-to-all connectivities, right? They sort of say, well, it's pretty close approximation of how the cortex works to say everything's almost equally likely to connect to everything. We can replicate a lot of what happens in the cortex using that. And so one idea may be that there's a lot of flexibility in cortical function based on this huge variety of connections. And in fact, there's already in this previous slide, whoops, oh, I'm getting mixed up between Zoom and PowerPoint, there we go. Right, that there is flexibility. So we're really interested in studying this flexibility. We love the idea of using these probes from NeuroNexus that are linear and have 64 sites on them. Actually, I think I might have grabbed one that we don't exactly use. I think they're usually on the edge. But the point is there's 64 sites distributed over the entire cortex and actually even a little bit below. And so we can really densely sample what's happening in the cortex among the cells. These sites are about 20 microns apart, which means that's about the size of a 20 microns apart vertically. So that's about the size of the cell body of a pyramidal cell. So if a neuron's active, we'll probably see it. And what we can do with that is functional mapping, right? So we can implant these chronically, and we can do some of the replication of these older studies where people are making one cell firing and seeing another cell fire. We can also, which is what I'm showing here, right, where we can try to look at functional connectivity between neurons, where one cell fires at this time 0, another neuron tends to fire 2 milliseconds later, or it tends to be inhibited 2 milliseconds later, indicating a possible synapse. We can look at those kinds of functional relationships in real time across layers. We can also do some of these more simplistic and really important fundamental questions of kind of which way do things spread. And then we can add, I'm going to skip this slide because I think I have a lot. We can add precision. We can add performance as well. Because I want to see how cortex is acting differentially and how the layers are acting differentially with performance. Because performance is obviously what we think these brain circuits are built for, right? To let us do things that are necessary, to let a rat do something that's necessary, and mouse do something that's necessary. So we're using mice. We're putting these electrodes across the layers of their cortices and having them perform a task while we do so, while we record. I'll introduce the task and briefly. So the idea is we're in somatosensory cortex, whisker cortex, and we're going to use a whisker-based stimulation. Basically, if we deflect the whisker one direction, let's say this sort of posteriorly, they're allowed to lick for water. If we deflect anteriorly, they're not supposed to be licking for water. And if they do, they have a little bit of a timeout and they don't get water for a little bit of extra time. So to maximize the amount of water that they get, they're gonna want to lick only when they're supposed to and not when they're not supposed to. So this is a go and no-go task. And we're using this rig that has a combination of head fixed recording so that we can give these precise stimuli, right? Because it's pretty hard in a freely moving animal to be stimulating its whisker with any precision. But we also want to do as best we can to look at kind of natural sleep both before and after stimulation. So this is where just a little bit of the tech comes in, right? Where we've developed a method that lets us record from the same animal, both freely moving and head fixed, by using a kind of a collar that's able to be locked into place. And so our head fixed rig, which will come into more play later, lets us do lots of things. It both lets us give these stimulations at the whiskers. It lets us monitor the eyes, including pupil dilation, to look at sympathetic tone, probably neuromodulatory tone, like the noradrenergic and cholinergic systems. And it also lets us monitor the whiskers as they move using cameras. Not to mention when the animal licks, how it's performing on the task. So the road we started down on, which I'm not going to be able to tell you the end of, but which I was kind of excited to start to share, is the fact that we're starting to study how these animals are performing and how their brain is responding as they learn this task over the course of 14-day training. This is something that hasn't been looked at, and it certainly hasn't been looked at across layers. So we have 14 days of 500 trials a day. There's an animal, a mouse, done this on basically 2 mice with nice recordings, where they're looking for water with this go, no-go task. And what we can see is that overall, there's just kind of this slow trend that they kind of tend to get better and better, more and more accurate over time. But if we look more closely, early in training on day 1, we can see, so we could focus here, for example, on the red and green lines. So we can see that at the beginning of the day of day 1, they're just kind of randomly licking, getting some hits, some false alarms. And then at some point they fall off and just basically stop licking. And they start getting this other kind of error where they're just sort of they're sort of omission errors in these dotted lines. So at the beginning of the day, there's this activated state. They're trying to get water. They probably get enough water and they stop. It's a high licking state. This is a low licking state. By the end of training, what we can see is that there's actually kind of 3 states. There's a high licking state. Now, if we focus on the black line, that's accuracy of performance. We see this peak in accuracy of performance around the middle of each day. We can actually see these here in our plots of performance over time as well, over the days. So every day we have this peak of performance And it's correlated with kind of, it follows an initial period where there's a lot of licking. So we have these solid lines up high, and there's either proper hits, or they're kind of licking too much, or alternatively, and then they kind of become accurate. And then later on, they stop licking and their accuracy goes down. So there's this kind of optimal period where they're both licking enough, but they're not overly satiated. And what we did was we plotted this over, took those same curves, essentially this black curve here, and plotted it over the 14 days. And so this is now 14 days of time, and this is those same black curves, but now in color. And you can see that these peaks of performance in the middle start to emerge kind of halfway through training, and they kind of stay. And what we're really interested in doing is trying to look at what are the correlates of that in the brain, right? Which layers are changing to allow that performance to work better? What are the predictors of that performance change? Let's say wheel running, how much, I didn't even mention that, but that's something we're measuring too. So we can see, for example, at the beginning of every day, they run a lot, they lick a lot. This is lick rate, this is wheel distance. Pupil diameter is actually changing over the course of learning because it's changing on this vertical axis. And if we go in and look at spiking, sorry, this Zoom thing is in the way. Okay, yeah, so if we go in and look at spiking, right, we have, this is our map of essentially correctness, and we have a somewhat similar map of spiking similarity from trial to trial. So this is something we're really starting to look at. One more data slide, I think, right, which is actually that, so actually just to crystallize one thing, we have this mixture of learning over time this way and brain state within each day, right? So to obtain high performance, the animal has to be both in the right brain state and has to, of course, have learned, right? So we have this mixture of ongoing moment-to-moment brain state and learning that we can look at the combination of across the layers. And so one of the things we've already been able to do is to look at the fact that, so we can just say early session, so one brain state where they're very licking a lot, to mid session where they're accurate, to late session, the brain state's changing. And we can see that actually what this is mapping is, on this vertical axis is the location of neurons. And we're basically just looking at modulation of different neurons. Each of these horizontal bars is a neuron being modulated either up or down on this metric of spike train similarity, which I could talk about if someone's interested, but sort of trial to trial similarity. And what we're seeing is that from early session to late session, we see that neurons are being up and down regulated. So these neurons are to the right in one case, right? They're up modulated, whereas in mid and late session, they're down modulated. And what's interesting is that they're very clustery across layers, right? Because this is vertical anatomy, anatomical location. So we're seeing that actually the neurons are tending to be grouped in terms of how they're modulated by which layer they're in. So these within day brain state changes are modulating the layers differentially. That's something we're excited to study. And we are starting to look, and then COVID came, and we had this kind of fortunate, chance to, it was obviously unfortunate, but we have in Michigan here, we have a very sensible governor who told us to stop. So we went home, and Lesio Bueno, who's I'm sure I'm messing up his name for anyone Brazilian in the crowd, but he's the postdoc who's doing all this hard work. And he went home and started analyzing his data and ended up making a discovery about REM sleep from this same data set. And so I thought what I would do is kind of introduce you to where we're going with this performance correlates across layers and across brain states. And that's where we're hoping to go in the long run. And then I thought I would also just take the chance to share with you and to give you some actual data, which I figure people always like, about another story that seemed to be emerging from the very same data set. And just to kind of introduce the importance of this, right? Actually, my postdoc, I was with Yuri Buzaki, and I was studying sleep. Sleep is crucial for optimizing and maximizing learning. So between days 1 and 14, obviously there's lots of sleeping happening. Lots of hippocampal and cortical researchers have shown that only with proper sleep does optimal learning happen, right? And so we actually think that what I'm going to show you now may eventually fold back into some of what we're going to look at later with learning. Anyway, so let's jump into it. So if we think about sleep, you guys have probably seen this, but there's the basic structure of sleep that people talk about. So, here's a person over the course of an 8-hour night. They might start out awake and then drop into deeper and deeper non-REM sleep. And they might every once in a while flutter up to these REM states and fluctuate back down to non-REM, up to REM, back down to non-REM. And so these REM states, right, are something less studied, and these non-REM states are more studied. So despite the fact that probably more people have heard of REM, probably because non-REM has a very uncatchy name, non-REM is first of all much more prevalent during each night of sleep and is much more studied. So you may have heard of things, like sharp wave ripples, K complexes, delta waves, spindles, slow oscillations. Those are all during non-REM and has been much better studied in terms of impact on sleep, especially spindles and ripples. But even slow oscillations for sure. Here we focus on REM, where a little bit less is known, actually much less is known, even to the point that we don't really know super much about even the structure of REM sleep itself, and kind of what are the implications for those ripples and spindles and things in non-REM, and if you stop them, you stop memory consolidation. What about REM? So what we're doing here is kind of looking at a typical way that a human sleep scoring person will try to look at REM. They'll take it apart into phasic and tonic aspects. So phasic REM is when the rapid eye movements happen in a person. So you can think of tonic as kind of this baseline state where not much is happening. It's just kind of low tone. And phasic is when something phasic interesting is happening. There's an event, right? So phasic is when the rapid eye movements are happening. Tonic is when there's kind of not much else. And that's been known for, this is far from new, but there's this kind of binary way of thinking about it. Oh, sorry. So the question is, do we see binary switching, right? If there are these phasic and tonic aspects to REM sleep and to how the brain is kind of going through this state, we should see something like a bimodal distribution of certain of the measures we take. I'm just looking at the time. I'm going to go quickly. We can find REM sleep using theta oscillations in the hippocampus, and so that's just the method we're using. Actually, pupillometry and sort of rapid eye movement hasn't really been able to be studied in rodents. Instead, we use the theta oscillation because we can record from their brains a lot more easily, and it's known that their whiskers twitch during the phasic part of REM. So we kind of went in, found REM sleep based on these theta oscillations. It turns out, actually, the reason we were able to get this is those same probes. I don't think I showed a picture of this. Those same probes that span the cortex, we actually found that they hit CA2 region of the hippocampus. And NeuroNexus worked with us to change the spacing actually from 20 to 25 microns so that we didn't have to be lucky to hit the CA2, so that we would basically in every recording get actually hippocampus and cortex in one. So all layers of cortex and hippocampus simultaneously. And so we took that and took some of the ideas from other researchers. There's a relatively, well, not too recent actually, I guess, study showing that phasic and tonic REM in rodents were separated by, so we have this theta oscillation, which is like an 8 hertz oscillation that's really prominent in rodents who are running around and exploring. It really organizes the spikes. You might have heard of place cells. It packages them beautifully into kind of, certain parts of the oscillation get spikes and certain parts don't, and they're organized in time. So this theta oscillation is really interesting and important in the hippocampus. And difference between tonic and phasic REM is these theta oscillations are changing their frequency from kind of low end theta oscillations around 6 hertz to higher end frequencies of theta oscillations around 10 hertz in phasic REM. Again, that's that kind of active, people are moving their eyes, animals are moving their whiskers, and this is the higher frequency, which correlates with when animals are running faster and being more active, actually the frequency goes up too. And so when we look in our own recordings, we can see that there's a general strength in the REM band, sorry, in the theta band. Malesio went and was able to kind of trace the peaks using some nice functions in MATLAB actually that let us trace peaks, and we used a few different versions of it and averaged them. And we're kind of comparing high theta periods during wake as well as kind of a control. And what we find is that it's not bimodal, right? We find that this theta oscillation, the oscillation of the frequency of theta that goes back and forth between tonic and phasic is not bimodal. Instead, there's a continuum between tonic and phasic. And if we take one of these and we see that there's our automatic sleep scoring, so red are these little REM incidents over this 9 hours, we can concatenate together a lot of these. These are actually at different time bases. We can concatenate all the REMs together, and we can see that there's a little bit of possible regularity of oscillation. And so when we go and analyze that, we see that actually REM sleep is switching between a high theta frequency and a low theta frequency at this something called infraslow frequency. So this is a new discovery that actually tonic and phasic are on a continuum and that they're oscillating with each other. And we'll jump ahead. We also did this in our, so that was all in freely moving animals. We can now do this also in head fixed animals and show that actually pupil, like eye movement, rapid eye movement actually is bimodal, interestingly, whereas whisker velocity isn't bimodal. So we kind of have this split where theta oscillation isn't bimodal, but on a continuum. Whisker velocity isn't bimodal, but is on a continuum, but actually rapid eye movement is. And that the rapid eye movement is mapped to different parts of this cycle of the theta frequency, right? So kind of at the peaks, there tends to be more rapid eye movement and whisker twitches. And so same thing, basically, quantification. And so what we showed here with these REM findings is that tonic and phasic REM are on a continuum with each other, at least by some measures. They fluctuate at infraslow rhythms. Infraslow is actually quite interesting. It actually has been found by some Italian groups to modulate performance. I bumped into it actually earlier in my career. It controls the structure of non-REM sleep too. So there's something going on there. It's actually what the fMRI people are looking at with resting state networks. So all the resting state network, that's infraslow oscillations. They're like about a minute long, 0.02 hertz. It's about 50 seconds. So there's something going on with this infraslow rhythm. It's controlling non-REM, REM, and waking performance. And it turns out, we were able to see this new finding in REM. And we actually, by the way, I don't think anyone has shown that eye movement is modulated in REM sleep in mice and rats the way it is in humans. So we're able to see that. And I'd love to see how these REM phases contribute to learning over time, along with the classical non-REM measures as well, when we come back to kind of having that full data set and our full set of questions. I want to thank the lab. This is Lesio, who did, I don't know, 98-9% of this. Mingxin Ding, she left the lab and is doing PhD at Brandeis, but she set up the rig, and then Lesio picked it up, kept setting it up, and has done all these amazing recordings. And with that, I'll stop. Don't want to do anything silly. There we go. And
thank you.
Happy to
Thank
take
you
questions.
very much, Dr. Watson. Fascinating. And who says COVID-19 has not?
Yeah. Yeah, if you get a little extra story out of it, it's not the end of the world.
Yeah. So I have one question here from Preston Donaldson. Are there any noticeable changes in depth LFP or spike phase coupling with performance in the go, no-go task?
Yeah, so that's exactly what we need to look at. You're thinking exactly the way we're looking at it. So we're looking at current source density, so kind of mapping of inputs and outputs along the layers. We're looking at spiking. We're actually working with a computational lab here at University of Michigan, Michał Zokowski's lab. So we're going to look at some spike rate vectors, both in terms of spike rate, actually I said spike rate vectors, but also in terms of spike timing regularity. So we want to kind of go to town and see what does or doesn't fall out. And I'm fine if we find that the cortex is modulated by layers, that's great. And if we find that it isn't, that's great too. So I don't know yet, but that's exactly the kind of direction we're trying to go in. So I shared a story that's not even a story yet, just because I thought it would be exciting to kind of talk about this rather than my postdoc stuff. Yeah.
So I'm looking to see if there are any other questions that have popped up. Please put questions in the Q&A so that I can ask the questions as much as I can. I know we are creeping into the discussion time that we have. One quick question, as you're looking at all this cortical layer stuff and all these oscillations that go from the continuum versus more discrete in the REM sleep part, do you have do you see the complexity of the layers producing these oscillations that might be reflected in
Yeah, so
Continuum versus non-continuum?
yeah, so well, we're still looking a little bit more at that. I mean, one thing is that we were focusing, the theta oscillations that we were focusing on are from the hippocampus because they're much clearer and they're generated in the hippocampus. But one thing that is known, for example, is that upper layers in the cortex versus lower layers are seemingly independent, for example, in the gamma frequency. So if you just look across the layers and you just say which electrodes are correlated with each other, the top set of electrodes will be correlated and then the deep layers will be correlated. And so there's a whole lot to explore there. I was surprised to see, I mean, that was just an exploratory analysis I did at one point as a postdoc. So I'm actually quite genuinely curious to see how much layers do or don't matter. I think they do, but it hasn't been clearly shown.
But there's two more questions. One is from Sarah Fauda. Hello, thank you for the talk. Can you speak more about the spike trend similarity factor and what it represents?
Yeah, so we actually did something a bit non-traditional just because Leslie was interested. So what he actually was doing was looking at trial to trial similarity using a vector approach. So he bins, so usually what people do is they'll make each neuron's firing rate be a dimension and then make a vector in that space to represent the firing rate, let's say in the firing rate in one trial, and then compare the vectors across trials. What Leslie decided to try doing was actually to make the bins of each neuron be the dimensions. And so this was a vector of what are the relative firing rates in time bins relative to the stimulus, and how similar are those to each other over time, and then he averaged across neurons. And it's just a very gross initial look at kind of how reliably are neurons firing from trial to trial. And we found that they go up when the animal's more correct, which is sort of not surprising. And now we want to dissect. That was actually what I was showing is modulated across state. That actually changes by state. And we've corrected for firing rate. So we turned all the vectors into unit vectors to take out any firing rate changes.
All right, we will do two short answers so we can go to the panel after that for discussion. And but the question is, what would bilateral / interhemispheric recording add to this story?
Your imagination's probably as good as mine on that. I feel that we know so little that I could speculate, but I don't have any concrete answers, and I was told to give a short answer. So I think I'll stop.
Oh, I'll ask this one last question for you. Igor Filipov asks, Brendon, we here work with infraslope brain potential oscillations. Have you attempted to record extracellular ISOs together with spiking in order to compare them from the same electrode site? Thanks for the nice talk.
To record extracellular together, yeah, we do extracellular together with spiking all the time, and that's definitely one of our next steps. Is so right now we're doing extracellular LFP only, just in these initial analyses, and we want to see how spiking is modulated by, in this case, REM or performance states, with lots to explore. For example, does phasic REM reflect previous experience more than tonic REM? That would be an example.
Okay, thank you very much, Dr. Watson. At this point,